Welcome to the Genomic Data Commons tutorial on the single-cell RNA-Seq analysis tool. The GDC's single-cell RNA-Seq tool can be used to analyze single-cell RNA sequencing data with cluster plots and gene expression overlays. To access the single-cell RNA-Seq tool, we will first load the GDC's Data Portal homepage at portal.gdc.cancer.gov. Then click on Analysis Center, which is the computer screen icon in the upper left corner of the web page. Under the Analysis Tools section, we will locate the single-cell RNA-Seq card, which is on the second row of Analysis Tools in this tutorial. Please note that the locations of the tool cards may change, so it is important to read the title of the cards. Once we have located the single-cell RNA-Seq card, we will click on the play button in the upper right corner of the file. The single-cell RNA-Seq tool allows users to select a sample associated with cases in the current cohort or all cases if a cohort is not created. If the active cohort is not of interest for this analysis, we can switch cohorts by clicking on the drop-down menu and selecting a different one at any time. If you need assistance in creating a cohort, please pause this video and use the Cohort Builder tool to create one. You can refer to the GDC Cohort Builder video for assistance. As the GDC is continuing to add single-cell data, this tutorial will be demonstrated with a previously created cohort of brain cases with single-cell RNA-Seq data. To start using the tool with a selected cohort, select the Single-Cell RNA-Seq tool in the Analysis Center and select a sample. A table of all samples associated with the cases in the cohort is displayed in a scrollable table along with descriptive information about the sample, including the case, sample, project ID, the primary site of the cancer, the disease type, and experiment ID. For this demonstration, we will select sample C3N0278401, which is a glioma tumor sample from the Clinical Proteomic Tumor Analysis Consortium, CPTAC. Once a sample is selected, 5 primary tabs are displayed, and you are navigated to the Plots tab. The 5 primary tabs are Samples tab for initial sample selection, Plots tab for dimensionality reduction, visualization, and population analysis, Differential expression tab for comparative analysis between clusters to find potential biomarkers, Gene expression tab for examining individual gene expression patterns across clusters, Summary tab displays the distribution of the expression of a particular gene of interest across clusters, along with pairwise comparisons between clusters and descriptive statistics. Each tab provides specific tools for data exploration and statistical analysis, enabling reproducible single-cell data interpretation. Let's review the Plots tab. The system automatically generates a Uniform Manifold Approximation and Projection UMAP visualization for the selected sample. Along with the UMAP plot, you can also select to view the T-distributed stochastic neighbor embedding (TSNE) and principal component analysis (PCA) plot by selecting the appropriate checkbox to the left of the Plots label below the Plots tab. You can select to view all plots or examine each plot separately. If selecting multiple plots, plots may appear next to or below each other based on your screen size and resolution. In this demonstration, we will select to view all plots. Plots display cellular relationships through dimensional reduction, with all cells shown as individual points on a 2D plot. Clusters represent distinct cell populations. The UMAP plot visualizes both local and global relationships and preserves population structure across scales. The TSNE plot emphasizes local cellular relationship, highlights fine population structure, and is optimal for detailed cluster analysis. The PCA plot displays primary sources of variation, reveals underlying data patterns, and presents distribution across components. You can also perform comparative analysis by visualizing multiple plots. For example, when comparing UMAP and t-SNE plots, you can look for similar cluster patterns, complementary structural details, and validate population identification. When adding the PCA plot, you can observe variant-driven relationships and can confirm population distinctions. The UMAP, t-SNE, and PCA plots include visualization properties, navigation controls, and settings and download options that are available in all plots. We will demonstrate each feature using the UMAP plot as an example. Let's start with visualization properties. Each point within the plot represents a single cell from the dataset. Point positions reflect closeness or distance in cellular similarities. Points closer together may indicate shared characteristics. Distance points may suggest biological differences. Color coding distinguishes cellular clusters. Each color represents a distinct cell population or subtype. Groups clustered together may indicate common or overlapping features. Navigation controls allow you to zoom and pan within a plot or set other controls. Zoom functionality. Zoom controls plus/minus magnifying glass icons are located in the upper left of the plot. Higher magnification reveals detailed cluster boundaries. Lower magnification shows global population distribution. The mouse scroll wheel can also be used to adjust the plot scale. Pan controls. Clicking and dragging within the plot repositions the view. Panning enables focused examination of specific regions. The zoom level is maintained during position adjustment. View reset. The reset button, circular arrow icon, in the left panel restores the default view and returns the plot to the original scale and position. Hide show cluster controls. Clicking on a specific cluster within the cluster legend on the right side of the plot presents options to hide or show specific clusters. By default, all clusters are shown. When selecting to hide a cluster, the cluster is crossed out with a line. By reselecting the hidden cluster, it is made visible. Showing only a specific cluster is also supported by selecting Show Only when clicking on a specific cluster within the cluster legend. When selecting this option, all other clusters are crossed out with a line. Clicking on a specific cluster again provides the option to Show All Clusters, resulting in all clusters being visible. Settings. Clicking the Settings icon, 3 horizontal lines, allows you to change the chart width, which controls the horizontal plot size, and height, which controls the vertical plot size. You can adjust the plot size in each dimension by entering the number of pixels in the text box. You can configure the dot size, which controls the cell diameter and opacity, which controls the transparency of each cell. To configure the dot size, enter a range between 0.01 and 0.1. Smaller values reveal fine population boundaries, and larger values emphasize individual cells. To configure the opacity, enter a range between 0.1 and 1.0. Lower values, less than 0.3, reveal density in overlapping regions and highlight population transition zones. Higher values, greater than 0.4, emphasize individual cell positions and define clear cluster boundaries. For our demonstration, we will first perform dense population analysis by reducing the opacity to 0.2 and decreasing the dot size to 0.01. This reveals gradient patterns in overlapping regions. We will then perform a rare population examination by increasing the opacity to 0.7 to 1.0 and increasing the dot size to 0.04 to 0.1. This highlights individual cells within a sparse region. Clicking the download icon, down arrow, on the left side of the plot beneath the settings icon, 3 horizontal lines, allows you to select to download the plot image in both PNG and SVG format. If multiple plots are displayed, images for each plot will be downloadable as separate files. The different dimensionality reduction visualizations serve as the foundation for subsequent analysis through the differential expression and gene expression tabs. Let's review the differential expression tab. Differential expression analysis calculates the top genes that are differentially expressed between clusters based on certain parameters. In the Differential Expression tab, a cluster can be selected to compare the genes that have been differentially expressed between the selected cluster and all other cells in the sample. For this demonstration, we will select cluster 11 from the pull-down list. Once a cluster is selected, a table filtered on the basis of both log fold change and the false discovery rate, FDR, is returned with the differentially expressed genes sorted by log2 fold change. A gene can be selected to display the expression levels across all cells in the sample. For our demonstration, we will select the gene CD247 by clicking on the radio box. A UMAP for the expression of the CD247 gene is displayed for the sample in the Gene Expression tab. We will be reviewing the Gene Expression tab later. Going back to the Differential Expression tab and selecting our cluster, Cluster 11, we will select the Gene Set Enrichment Analysis subtab. GC allows us to examine the pathways that are most enriched in samples after a differential expression analysis is run. Various options can be selected to refine the analysis by clicking the Settings icon, 3 horizontal lines, in the left panel. Number of permutations, a higher number gives higher accuracy, but with a longer runtime. Minimum and maximum gene set size filter to control the size of the gene sets used in the analysis. Filtering non-coding genes from the analysis. Choosing a cutoff parameter controls whether FDR, or top gene sets, is used as the cutoff for gene sets. Specifically, the cutoff method allows the user to set a specific number of gene sets ordered by their ascending FDR value. Gene Set Group Selector allows selecting from multiple options, including those from the Gene Ontology, Reactome, Wiki Pathways, and Hallmark gene sets. Once a gene set group is selected, a rank plot is displayed. We will select the Gene Set Group H, Hallmark gene sets. Once selected, a table is displayed below the Gene Set Group selection. The table includes the gene set, a normalized enrichment score, the gene set size, p-value, and FDR. The table can be downloaded by selecting the down arrow to the upper right of the table. You can use the radio buttons in the table to select a specific gene set. We will select the first gene set, Hallmark Allograft Rejection. Once selected, a rank chart for the gene set appears above the table. The rank chart visually represents how genes within a specific gene set are distributed across a ranked list of all genes based on their differential expression. To investigate gene expression across cells, click on the Gene Expression tab. You can initiate a gene query through the search field in the upper left. The search field provides dynamic suggestions based on input. We will demonstrate a gene search using the TP53 tumor suppressor gene. Following gene selection, the plot updates to display expression levels of that gene across the cells in the selected sample in a UMAP plot. To the right of the plot is a legend that provides a color gradient for gene expression levels. Darker red indicates higher expression levels. By clicking on the color bar in the legend, you can configure the color of the expression legend through the minimum and maximum selectors. For both the min and max, the tab presents a color selection window enabling precise control of the color and hue that are selected. You can also select automatic, fixed, or percentile in the pull-down list above the min and max color selectors to specify the method for setting min and max. Clicking the Settings icon, the three horizontal lines in the left panel after entering a gene in the Gene Expression tab, allows you to configure the plot and dot size and show and hide the plot grid, which I've previously demonstrated. For the gene expression plot, you can also choose to show unexpressed cells or change contour features in support of density analysis. You can set a variety of contour map parameters once selecting to show the contour map. The Show Contour Map checkbox toggles the density-based contour map. The contours are weighted by the gene expression values of each point. This allows the contour to visualize regions of high expression. The default is to have the contours turned off. Contours can be enabled by checking the Show Contour Map checkbox. For our demonstration, we will enable the contour map for the TP53 gene for our selected sample C3N0278401. Once the contour map is enabled, you can enable colored contours via the Color Contours checkbox. By selecting the checkbox, you can control the resolution of the contour map by entering a value in the Contour Bandwidth text box. The default value is 15. Smaller values capture more data variation and therefore make a more detailed map. Larger values smooth out the lines by taking into account a wider range of data and focusing less on the nuances of the data variation. You can also control the density of the coloring of the contours. The default threshold value is 10. Smaller values make the coloring less dark. Larger values make the coloring darker. There are a variety of analysis applications for visualizing gene expression in the contour map, such as population center detection, transition analysis, and rare population identification. Population centers can be detected when dense regions appear with concentrated contour lines. This reveals primary cluster locations and identifies population boundaries. Population transitions are apparent when there is gradual contour spacing. This highlights regions between distinct clusters and reveals subtle biological state changes. Rare populations can be identified when there are sparse contours. This indicates low-density regions, helps locate isolated cell populations, and distinguishes rare cell types from artifacts. Let's review the Summary tab, which allows you to see how highly differentially expressed genes vary across clusters. When selecting the Summary tab, a violin plot is displayed, showing the distribution of the gene of interest across all clusters in the sample. When mousing over a cluster in the violin plot, measures of central tendency such as mean, median, variance, and others are provided. The table to the right of each cluster shows pairwise statistical comparisons. This comparison is performed between all clusters for the particular gene of interest, with their respective p-values generated by the Wilcox-Rank sum test method. Clicking the Settings icon, the three horizontal lines in the left panel after selecting the Summary tab, provides several options to refine the violin plot visualization, including the ability to alter the orientation of the plot, horizontal or vertical, symbols to represent data, ticks or circles, and the symbol size. Scale for the axis of the plot, linear or log 10. Visualize data as either discrete or continuous. Other options, like the ability to change the plot length, thickness, and padding between each violin in the plot, and to modify the color of the violin plot fill, are available. In addition, there are options to control the number of bins used to make the violin plots and the length and width of the median symbol. To exit, we can click the X button to the left side of the single-cell RNA-Seq header, which will bring us back to the Analysis Center. For more guidance on this and other GDC data portal tools, please refer to the video tutorials or go to docs.gdc.cancer.gov.